Hello my students, I'm your biology teacher, Shekhar Govin. Our topic today is sexual reproductions in flowering plants, and our subtopic is 6.2, developments of pollen grains and embryo sex. Learning standard number 1, describe the formations of pollen grains in an anther. Number 2, describe the formations of embryo sex in an ovules. The formations of pollen grains in an anther. The part of the flower that produce pollen grains is the anther. So this is anther, okay. How are pollen grains produced? Diagram below show developments of pollen grains. So this is lobes. So this is filaments, okay. And then this is the real diagram, show the anther and the filaments. Okay, both of these we call it stamens. This is the male reproductive organs for flowering plants. Anther produce spore that contain the male gametes. So the male gamete, we call it pollen grains. Okay, this is another diagrams. This is anther. The whole part is anther, and one part of this we call it pollen sacs. Okay, this is filaments. This is lobes, and then this is microspore, mother cells, which is diploids, and this is then another diagrams, pollen sacs, and this is microspore, mother cells. The anther is usually composed of 4 pollen sacs, okay, 1, 2, 3, 4. During the development of anther, a group of tissue grow inside each lobes to form 4 pollen sacs, 1, 2, 3, 4. Okay, in each pollen sacs, there are hundreds of pollen mother cells called microspore mother cell, which are diploids. Okay, this is the pollen sacs, and this is the microspore mother cell, which is diploids. Okay, diploid means complete number of chromosomes. Stage number 2, the microspore mother cells divide by meiosis to produce 4 haploid microspore cells. Okay, 1, 2, 3, 4. So these 4 microspore cells are collectively known as tetrads. Each cell in the tetrad develops into a pollen grains. Okay, each cell, okay, one of the cells, and then form the pollen grains. And then the nucleus in the pollen grain divide by mitosis to produce 2 nuclei, namely the generative nucleus and tube nucleus. This blue color is the generative nucleus, and this one is the tube nucleus. The wall of the pollen sacs, which is thick and waterproof, break when the pollen grain matures. The pollen grains are released. Okay, this is another diagram, show the formations of pollen grains in an anther. So this is anther, okay, and there are 4 pollen sacs here. So each pollen sacs, there are hundreds of microspore mother cells, which is diploids, and this microspore mother cell divide by meiosis to produce tetrads. Okay, each cell in the tetrad develop into pollen grains. Okay, and then the nucleus in the pollen grains divide by mitosis to produce 2 nuclei, namely the generative nucleus and the tube nucleus. Okay, tube nucleus and generative nucleus. Okay, this one is the same diagram like just now, but this one is the actual specimens. Okay, from this diagram you can see this is microspore mother cells, which is diploid, and this is meiosis process, and this is microspore cell, polytetrad. Okay, and then this is the pollen grains. Okay, the nucleus in the pollen grain divide by mitosis to produce 2 nuclei, which we call it generative nucleus and tube nucleus. Okay. Activity 6.2. The aim of this activity is to prepare and observe the slide as well as describe the shape of pollen grains of various plants through a light microscopes. And this is the material: enter of various type of flower and 3% sucrose solutions. The apparatus: forceps, light microscope, cavity glass light, cover slip, and dropper. The procedure: number 1, add a few drops of 3% sucrose solutions to the cavity glass slide. Okay, you can see from figure 6.4a. Okay, these ones. And then number 2, obtain some pollen grains from a matured enter by using the forceps. Drop the pollen grain onto the 3% sucrose solutions. Okay, these diagrams. This forceps drop the pollen grain into this cavity glass using the forceps. Number 3, place the cover slip over the cavity on the slide and leave it aside for 3 minutes. Observe the slide under the light microscope using a low power objective lens, and then repeat step 1 to 5 using pollen grains from different plants. Draw a diagram of the observed structure of the pollen grains. Discussions, questions. Why does this activity use cavity glass slides and 3% sucrose solutions? The answer is the answer for A, to produce a 3-dimensional image of pollen grains. Okay, that's why we use cavity glass slides. And why we use 3% sucrose solutions? 3% sucrose solution is a solution that is isotonic to the pollen grain cell. So these solutions can maintain the shape and size of the pollen grains. Okay. Number 2, based on your sketch, state the names of the plants, the feature of the pollens, and the importance of this feature. Okay, this is based on your sketch. This is hibiscus, spiky with hook. The important is easily sticks to insect legs and surface of stigma for pollinations. Okay, this one is Mimosa pudica. Feature of pollen grains: sticky, small. The importance: easy sticks to insect legs and surface of stigma for pollinations, the same like the hibiscus. And this one is lalang, thins in a large quantity. Okay, transfer easily by wind for pollination. Okay, that's why it's thins. Okay, this is example of insects pollinated flower. This A. So hibiscus is pollinated by insects, while the lalang, this is lalangs, pollinated by winds. Wind animals and insects are pollinating agents that help in transferring pollens. Small, smooth, and light pollens are transferred by winds. Examples of wind-pollinated flower are corns, grass, and paddy. Pollens that are carried by animals and insects are rough and sticky. Examples of animal-pollinated flower and insects-pollinated flower are rambutan, durian, papaya, rose, sunflower, and hibiscus. The formations of embryo sac in an ovules. Ovule are structure of flower formed inside the carpels. Ovule develops from a layer of tissue inside the ovary. Okay, this is ovary. Inside ovary is ovules. A single ovary may contain one or more ovules. In this diagram, only one ovules. The ovule attached to the ovary wall through a stalk called funicles. The area of attachment of the funicles to the ovary is called the placenta. Okay, the placenta supply nutrients to the ovule through the funicle. Okay. A mass of tissue inside the ovary develop forming a lump called nucleus. Okay, this is nucleus. Okay, the nucleus consists of parenchyma tissue. The nucleus tissue developed into two layers called the integuments. At the end of the integuments, there is a little openings called the microphyles, which allow the entry of air and water into the seed during germinations. And last one of the nucleus cells is the megaspore mother cells, or also known as the embryo sex mother cells, which will develop to form an embryo sex. Okay, this is the whole structure of embryo sex in the ovules. The formations of the embryo sac in the ovules. Number one, the megaspore of mother cells divide by meiosis to produce 4 haploid megaspore cells. This is haploids. This is megaspore. Okay, there are 4 here. And then 3 of the megaspore cell degenerates, and only 1 megaspore develops. Once develops, another 3 is degenerates. Okay, and then the nucleus of the cell that has developed divide mitotically 3 times to produce a cell with 8 nuclei. Okay, from 1 divide by mitosis 3 times become 8, and then the 3 nuclei move to the end of the embryo sex to form 3 antipodal cells. Another 3 nuclei move to the opposite ends of the embryo sex and forms 2 synergid cells and 1 egg cells. 2 nuclei in the center of the embryo sex form the polar nuclei. Okay, 3 become antipodals, 2 become polar nuclei, 1 become egg cells, and 2 become synergid cells. Okay, this is another diagrams. Okay, this is embryo sex mother cells. Okay, undergo meiosis process to form 4 megaspore, call it tetrads, but only 1 develops, the other 3 degenerates. So one of these divide by mitosis 3 times to form 8 nuclei, 3 become antipodal cell, 2 become polar nuclei, 1 become egg cells, and 2 become synergid cells. Formative practice 6.2. Formative practice 6.2. Name the cell that differentiated to form pollen grains in the anther. B. Embryo sac in the ovary. Okay, in the pollen grains, we call it microspore, mother cells. This is for males. And then B. Megaspore, mother cell. This is for females. Number 2. In the formations of pollen grains, the nucleus in the pollens will divide by mitosis to form 2 nuclei. Justify. Okay, produce 2 nuclei, namely the generative nucleus and the tube nucleus. The tube nucleus form pollen tubes, whereas the generative nucleus divide mitotically in the pollen tubes to form 2 male gametes. Okay. Number 3, what is the importance of meiosis in the formations of pollen grains and cell of the embryo sex? To produce gametes which are haploids. Okay. Number 4, after the formations of embryo sex is completed, this cell contains 8 nuclei. Name the nuclei. 3 antipodal cells, 2 polar nuclei, 1 egg cell, and 2 synergid cells. Okay, that's all for today. Thank you. See you in next lessons. Bye-bye.